Okay, we still haven't started yet, but I started the recording. Today is October 26th, coming up on Halloween weekend. All Hallows Eve is followed by the day of All Hallows, which is also known as All Saints Day, and the day after is All Souls Day. Day of the Dead. Okay, we're coming up on 10:30 here, so I'm going to take an image from the screen for my attendance. Comes 1, a 1, and a 2, and a 3. Then I'm going to take another one of the other screen. 1 and a 2 and a 3. Okay, Miss Rachel, are you ready to make your announcement?
Oh yeah, I'm good. Okay, so the PMA has a meeting a week from today. It's 5 p.m. in Le Bijou Theater in the Union. And November is a very busy month for PMA. The weeks after that, we have physicians coming speak. So be on the lookout for those. I'll send the graphic on Thursday because I don't know how to do that, but I'll figure it out before Thursday. And then yeah, so first meeting is this a week from today, 5 p.m. in Le Bijou Theater, and that's it.
Okay, thanks for that. If you haven't been around campus a while, Le Bijou Theater is kind of hidden in the student union. It's over there by the restrooms, by the front doors of the student union. Okay, Miss Lauren's reminding you that you might be able to attach a little flyer into the chat. Yeah. Or if you print it out, you're welcome to hand it out to the class or something like that. Okay, we're going to start class now. Like I said, this is Tuesday, October 26th. What we've been talking about is how to move the blood around. All right, we've gone over heart structure, heart anatomy. We went through that at a comparative level, looking at several different vertebrate hearts and even something like the heart of a crab. And today we're going to continue to talk about anatomy first, but we're going to transition to the respiration side of the equation. So the circulatory system and the respiratory system really work together because most of what the circulatory system is doing from second to second is it's delivering oxygen. It delivers other things that are important, like cell signals, like we went over in the past few weeks. You know, it delivers hormones like insulin. You need the circulatory system for that, but delivering oxygen's more important from second to second. It also delivers nutrition, nutrients. After you digest food, it's the blood system that moves around the nutrients, but from second to second, it's much more important to get the oxygen moving around. Okay, some of the concepts we're going to cover today are in the textbook on page 593, 601, 602, 608, 617, 623, and 648. These are covered in chapters 23 and 24 mostly. All right, we're going to start off talking about things, kind of big picture ideas. All right. So there's a variety of different organ systems that are made for respiration. And especially even in vertebrates, in our group, vertebrates, there's animals that use gills and there's animals that use lungs. Okay. Now, these organs are analogous because they help do the same thing. Analogy means they're functionally similar. So it's true that they're functionally similar. What happens with gills is blood comes to the gills and the cells in that blood are able to take oxygen away from water. That's analogous, functionally similar to what happens in the lungs. What happens in the lungs? Blood comes to the lungs. Blood is able, with its red blood cells, to take oxygen from the air, right? So these two very different organs have been adapted for this job of harvesting oxygen from two different mediums. Now, originally, our oldest ancestors that we come from were water-breathing ancestors, like fish. Okay, so in a lot of respects, this is an older architecture and kind of an older process. What's tricky about water is water cannot hold as much oxygen as air, right? This kind of makes sense. But water does hold oxygen. You can dissolve oxygen in water, but there's a lot more oxygen available in air. So one of the things that occurred as our ancestral animals, something like an early amphibian, came out of water and started breathing air, one thing that was allowed is a much easier way to harvest oxygen. Okay. Okay, we're going to talk more about this. So first, let's talk about these two very different systems to harvest oxygen. We're going to go in two very far extremes. And part of this story is that we accept that these two very different extremes come from a similar ancestor. So a very long time ago, we moved into a group of animals that are protostomes. They make their mouth first at gastrulation, and deuterostomes. That's us. We make our mouth second at gastrulation. And a nice example of a protostome is an arthropod. Okay, this is from high school and freshman biology. What's examples of arthropods? Those are insects and spiders and crustaceans. So a nice example of an arthropod for us is a blue crab. Okay. Among arthropods, some of these insects, like some beetles, move pretty slow, but blue crabs are kind of like lean, mean swimming machines. So they're almost like at a high extreme of an arthropod that can get things done. They're very active predators. They swim fast. A blue crab is made to swim, whereas a fiddler crab is not made to swim, right? They're walkers. But blue crabs are able to do pretty high metabolism in the water, right? Now, what they use in order to get oxygen is they use a type of gill because they're harvesting oxygen from water. But what flows through that gill is not usually called blood. In arthropods, this is called hemolymph. right? Like in conversation, you can say that something like an arthropod has something like blood, but generally we don't call it blood. In deuterostomes, vertebrates, and humans, we call it blood. But this medium that flows throughout the body is slightly different. So blue crabs and other arthropods, they use a biochemical pigment. We call it a respiratory pigment. Respiratory pigment that allows them to harvest oxygen from water. It's in their hemolymph, and that respiratory pigment is called hemocyanin. Right? It's not in a blood cell. It's loose within the hemolymph. So a term for that is extracellular. Since it's loose within the hemolymph, sometimes it creates polymers. Sometimes it can be single subunits working. So it can exist as a large polymer. Let's call it a large polymeric molecule. That means many different pieces of hemocyanin can come together. And in order to bind oxygen, it uses a metal. So before it can bind oxygen, it binds to a metal, but here, instead of iron like us, it binds copper. Right, this is copper, Cu. It's not calcium, which is another divalent cation. This is copper, Cu. Whereas hemocyanin binds copper, our respiratory pigment binds iron. That means blood is usually red, but because hemocyanin and hemolymph contains a binding property to copper, this whole pigment, as well as the color of the hemolymph, has a blue tinge, right? When you collect hemolymph from a crab and hold it up to the light, it's not blue like royal blue, but you move it around and you see, oh yeah, there's like a blue tinge here. Okay, this seems very different than what we use as vertebrates and deuterostomes, yet we know that this whole system evolved from a common ancestor, right? In some ways, to me, it seems like this is such a strong variable. It digresses so much from what we have. It almost seems like it comes from another planet, but it represents really many millions of years of having to do the same thing that we do. That is, it represents just another pathway to sort of design a respiratory pigment that can bind oxygen and take it away from water. Okay, in our group, in deuterostomes, we had to design a similar pathway. You call it analogous. It's similar, but there's hardly anything that makes it homologous. Doesn't seem like the molecules are similar. So we think that they evolved through a totally different pathway, right? So our example here for a deuterostome is going to be the human. And okay, we use lungs, but fish are also deuterostomes and they use gills. So the respiratory pigment that we use that's red is hemoglobin. Whether you're a fish or a human, we all among vertebrates use hemoglobins. Now, hemoglobin is not a large polymeric molecule. Instead, it's very specific that it has 4 subunits. So it's called a tetramer. It's not a variably sized polymer, it's a tetramer. It always works with a group of 4. And It absolutely does not bind copper. It binds iron. And that is what makes it have a red tinge. Okay. So we've been mostly concentrating on how humans and vertebrates get circulation done and respiration done. But we're also comparing that to these faraway cousins, crustaceans, which are a model for arthropods, where their circulatory system is pretty weird. What do I mean by it's weird? We talked about that last time. They have a neurogenic heart. Their neurogenic heart is a heart that beats, but it doesn't have the pacemaker within the heart. It relies on a ganglion very close to it to send it a signal to beat, right? That's the neurogenic heart. Its circulatory system is also called an open circulatory system. Now, it's not open to the environment, but it's open within the body of the crab. The heart beats, it sends hemolymph into the what you could call the hemocoel. It's like the body compartment of the crab, and it like bathes the muscles. The muscles are moving in this crab, and that sends the hemolymph back to the heart. So it's kind of open to the tissues. It's an open circulatory system. On the other hand, us, we have a closed circulatory system. We have a myogenic heart. Myogenic heart, it means the muscle of the heart itself has the pacemaker in it. And it's closed circulatory system. It's like a rule of our circulatory system that the red blood cells and the blood itself, it does not leave the circulatory system. Okay, you go from the heart to the arteries, to the veins, back to the heart. In a normal situation, that blood never leaves any vessels. It stays within the pipelines of the vessels. Okay, that sets up, a pretty nice exam question where you can compare and contrast. How do these compare? Well, they compare functionally because they both have to do the same thing. They have to move a fluid around a body, harvest oxygen from the media, and deliver that oxygen to the tissues. All of that's comparable, but they contrast. It appears that they evolved independently of each other because the respiratory pigments used by both are pretty different. The pigment holds on to copper in the crab, holds on to iron in the human, gives it different colors, and it's in a different place. All right. Maybe the final thing that I forgot to mention on the human side is instead of being extracellular, the hemoglobin in a vertebrate is always within the red blood cell. So it's not extracellular like a crab, it's always intracellular. Now, the red blood cell of a human is kind of weird because it has no nucleus. It's not a cell that just exists without a nucleus. It starts out with a nucleus when it's still in the bone marrow, and it has to kind of get rid of its nucleus. So it's a little trick that it does. It gets rid of its nucleus so that it can kind of like optimize its job of just being totally packed full of hemoglobin. And that means it can bind a lot of oxygen. All cells need a nucleus, but for the job that red blood cell doing is in our body, just towards the end of its life, gets rid of its nucleus so that it can really bind a lot of oxygen. There's other vertebrates like fish and frogs that never enucleate their red blood cells. They keep the nucleus in their red blood cells. So that's a reminder that kind of like the normal way to be a red blood cell is to have a nucleus, but mammals have gone a step towards the extreme to get rid of the nucleus. Okay, let's talk about these respiratory pigments. as I mentioned, in invertebrates, the main respiratory pigment that we use is hemoglobin. And hemoglobin, when it's working, is really made up of these 4 proteins that come together as subunits, right? You could think of them as kind of coming together like this. There is an alpha subunit, a beta subunit, another alpha subunit, and another beta subunit, right? You could call this alpha 1, alpha 2, beta 1, and beta 2. Okay. In the past, we've talked about dimers. Okay, a dimer is just 2 subunits that come together. This is a tetramer. It's 4 subunits that come together. And a lot of the function of hemoglobin is manifested through this particular kind of structure. So this is an example of a structure function relationship. Okay, these are the names. And I'm drawing these very loosely, but you know, these are protein molecules. Protein is like a chain of amino acids, right? So you could think of this as kind of like these ribbon structures. And what all of these hemoglobins have is they actually have a binding site for kind of like a cage in the middle. And that cage, you could call it kind of like the heme group. And molecularly, a heme group is a type of porphyrin. Okay, this heme is not part of the protein. It's this extra group that sits right here in this pocket. And it's the heme group that binds on the iron. So in the middle of each one of these subunits, there's kind of like a cage. I'm drawing this. I know it just looks like something like a wooden pallet or something like that, because it's kind of flat and it sits in the middle there, and it's surrounded by all these protein residues. Each one of these binds iron. So before binding oxygen, the protein has to have these other groups to it. It's a protein sort of structure bound to a heme. Heme is bound to an iron. And then these molecules, these subunits, can each bind one oxygen. Okay. So you can think of this one as binding an oxygen. This one is binding an oxygen. This one is binding an oxygen. This one is binding an oxygen. So the tetrameric structure of hemoglobin allows for the binding of 4 molecules of oxygen. And I feel like when you start thinking about the function of hemoglobin, the first thing you think of is it binds oxygen. Okay, it's a respiratory pigment. It's evolved to bind oxygen. Now, when I say it evolved to do that on purpose, you know, that's a little bit like slang, because you don't want to make it sound too teleological, but the evolutionary pathway that led to this tetrameric structure has allowed for the binding of 4 oxygens. But what's just as important in hemoglobin really is to bind oxygen and then release it, deliver it. Okay, we often don't think about that part of it, and it's not like binding is good and losing is bad. It's like it's part of the job. Hemoglobin is made to go to the lungs, grab oxygen from the air, and then go to the tissues, like to your pinky toe, and deliver the oxygen. Bind, deliver, bind, deliver. So it ends up being, its binding is important, but also being able to let go is just as important. And the structure function relationship is important for that. It has a certain structure when it binds, and it has a certain structure when it releases. Okay. Right. One way to sort of appreciate this is to understand that in vertebrates, there's a hemoglobin that has a tetrameric structure like this, but invertebrates, in mammals and other vertebrates, there's another kind of heme protein, and it's myoglobin. Myoglobin is related to hemoglobin. It also binds oxygen, but myoglobin exists, as you can imagine, because the word myo, it exists more in the muscles and in tissues, and it's more stingy than hemoglobin. And it exists only as a monomer, right? But it also has this protein support, and it's got a heme group in the middle of it, and that heme group binds on the iron, and this molecule can work as a monomer and grab on the oxygen. Okay, so what's the difference between the way myoglobin works and the way hemoglobin works? What this comparison is going to allow us to do is to sort of understand what's more important about this structure function relationship with hemoglobin. All right, so I'm going to set this up right now. And one way to talk about how these differ is to look at their oxygen saturation curve. Okay, and these oxygen saturation curves that are important for understanding hemoglobin function, what you usually see on the x-axis is the concentration of oxygen in the fluid, right? Now, we don't call it the concentration of oxygen exactly, because when you talk about something as a concentration, that's usually like a solute and a solvent, like salt. You know, salt's a powder. You can dissolve salt in water, and you can call that a solute and a solvent, and it has a concentration. But since oxygen is a gas, it is soluble in water, but instead of concentration, we call it a partial pressure. So I can say that what I'm plotting here is the partial pressure of oxygen. Let's say the partial pressure of oxygen in the blood. All right. How much oxygen is in the blood? So in kind of informal conversation, you can say how much oxygen is in the blood. You can say the concentration of oxygen in the blood. Nobody's going to call you on that. But physiologically and on the exam and in the textbook, we use this small p here to represent the partial pressure of oxygen. Okay, and over here on this y-axis, you could say that we are plotting the saturation of oxygen in the blood, oxygen saturation. So it's similar to think of this as the binding of oxygen in the blood, how much oxygen is bound up to hemoglobin. Okay, so if we plot PO2, we start at something like 10, 20, 30, 40. Because this is a gas, the units are millimeters of mercury. It's a pressure, the partial pressure of oxygen. All right. And we plot how hemoglobin works here. This is tetrameric hemoglobin. You know, it's got 4 different binding sites because it's got 4 different subunits. At a real low PO2, there's real low oxygen saturation. So if there's not a lot of oxygen in the blood, the hemoglobin is not bound up to hardly any oxygen, right? Now, if this was kind of like a normal molecule, you would expect, okay, the more oxygen you get, the more binding you get, right? That's kind of like what you would expect from something that is linear, but that's not how the tetrameric shape of hemoglobin works. At this real low PO2, we say that hemoglobin is in a tense state. And eventually, we're going to say that it goes from a T to R state. T stands for tense, R stands for relaxed. So one way that people describe this is like, at first, that tetrameric structure of hemoglobin is like real tight, especially at a low PO2. But as the PO2 increases, that structure of hemoglobin, it kind of begins to open up. It begins to relax. So this is why I said there's a structure function relationship. You know, you could say relax, you could say there's a slight conformation change as PO2 increases. And what we find is, as the PO2 increases, you can go up to something like this. Now, if we are looking at oxygen saturation, what we can be plotting here is 100% saturated to 50% saturated to something like 0% saturated. Okay, 50% of the molecules are saturated with oxygen, 100% of the molecules are saturated with oxygen. So one of the numbers that we use to describe this is that the P50 for hemoglobin here is, it would fall at around something like 28. That's the P50. That's where 50% of the hemoglobin is bound up with oxygen. So you would say the P50 is 28 millimeters of mercury. I can't see that. The P50 of this hemoglobin is 28 millimeters of mercury. And what has happened here is, it was kind of hard to get this molecule to open up at a very low PO2, but as the PO2 increased, one oxygen jumped on, and then another oxygen jumped on even more easily. And so the shape of this curve here is why we say that there's a cooperativity. There's a cooperativity in the structure function relationship of hemoglobin, in that as the molecule binds one oxygen, the subunits kind of cooperate so that it's easier and easier to bind the second oxygen, and then the third oxygen, and then the fourth oxygen, right? So this cooperativity has to do with the transformation from a T to R state. So you could think right here, it was hard to bind that first oxygen, but it was much quicker to bind the second oxygen, and then even quicker to bind the third oxygen. And then finally, you get 100% binding by the time you get to the top here, right? So if you look at this curve, rather than being a straight diagonal, people describe this curve as a sigmoidal curve. And the word sigmoidal simply means this binding coefficient curve, or this saturation curve, is S-shaped. Sigmoidal means S-shaped. There's an S-shaped curve, right? So that's how hemoglobin works. That's kind of like the more popular respiratory pigment that we think of as working from second to second. Well, how does that differ from myoglobin? Okay, myoglobin is in the muscles, and it tends to be more stingy. In a way, one way to describe that is, it doesn't deliver oxygen until the muscles really start running out of oxygen. All right, so I'm going to draw you a saturation curve for myoglobin. It's not sigmoidal. Instead, it doesn't bind much at a very low PO2, and it doesn't bind much as PO2 goes up. It doesn't bind much, it doesn't bind much. Then boom, it binds, and it's very unwilling to bind anymore. Okay, so it's got not a sigmoidal shaped curve, but instead a curve that gets bound up with oxygen at a very low PO2 and it remains saturated in all of these other higher PO2s. Okay, so there's kind of like the first way to think about this is that it takes hemoglobin longer to bind oxygen and then become saturated. Okay, myoglobin, it's quicker to bind oxygen, right? You might think that it has a very, it's easier for myoglobin to bind, okay? Because it only needs to be hitting a PO2 of something like 5 here, boom, it's bound. So one way to do that is to describe the P50. So you come over from the 50-degree mark here. Look, what's this P50? This P50 for myoglobin is like 5 millimeters of mercury. So one way mathematically to compare the binding or the saturation curve of hemoglobin to the saturation curve of myoglobin is to say, oh, the saturation curve for hemoglobin has a P50 of 28. Saturation curve for myoglobin has a P50 of 5. This is hemoglobin. But it really means two things. Another way to think about it is what's going on in the tissues, right? So if you think about what hemoglobin does, hemoglobin is moving through the blood, right? It's going through the heart, and when it gets to the lungs, it's at a place where there's a very high PO2. At the lungs, there's plenty of oxygen available in the blood. So at the lungs, it binds oxygen. We're talking about hemoglobin now. But as hemoglobin goes to the tissues, you know, the way that I just introduced this to you was that, okay, it's hard for it to bind, but really the same thing when I'm saying it's hard for it to bind describes what hemoglobin does at the tissues where there's low PO2. And what hemoglobin does is it delivers, right? A while ago, I told you it was hard for it to bind, but really what that's saying is like at tissues, it's delivering. So hemoglobin works this way. Hemoglobin picks up oxygen here. You could say it's harvesting oxygen. And here at the tissues, it delivers, right? So it's binding strong up here at a high PO2, but it's binding weak here at a low PO2. So it's picking up at the lungs, delivering to the tissues, picking up at the lungs, delivering to the tissues. Whereas, what does myoglobin do? Myoglobin holds on to oxygen at this high PO2. As you go to a little bit lower PO2, it's still holding on. As you go to a lower PO2, it's still holding on. As you go to a lower PO2, myoglobin, it's stingy. It's holding on, holding on. It doesn't release until the PO2 of the blood is real low. Finally, right here, it releases, right? So that means hemoglobin is kind of working for me and you when we're at rest. It's picking up oxygen, delivering oxygen. Whereas when we're sitting at rest, myoglobin is just holding on to oxygen and not letting it go at all, because the PO2 of our blood never gets real low like this. When we're at rest, the PO2 is always sitting here in kind of a comfortable high amount of oxygen. But maybe if you run a mile, right? You run a mile, your hemoglobin is working here, but you run a mile and there might be places in your tissues where you are starting to run out of oxygen. And that's when myoglobin says, okay, I'll give you some oxygen. So it's almost under muscular stress when myoglobin finally delivers. It's stingy. It waits until the very last bit of oxygen at a very low level of PO2. All right. So that's some of the structure function relationship between how hemoglobin works and how myoglobin works. Now, there's something else about this curve that I want to show you about hemoglobin. has to do with some of what we were just talking about. Like you could imagine hemoglobin working at rest and at exercise. And that is that the saturation curve of hemoglobin can change according to pH as well. It can shift in a different pH environment. Okay. So a typical pH, you could find this on page 646. I think there's a picture of a nurse on page 648. pH is 7.4. In a normal resting situation, in our blood chemistry, it's not sitting right at 7.0 like water would be for pH, but our blood is slightly more alkaline than that. It's 7.4, right? As we begin to exercise, we start to use up oxygen. We start to have more CO2 in our blood, okay? And also lactic acid kind of builds up in our tissues too. And so what you see in a exercise situation is you don't see a big drop in pH, but it drops a little bit. It goes to 7.2 during exercise. No, yeah, exercise. So it becomes slightly acidic, right? In an acidic pH, there's a shift of this saturation curve. The curve still has the same shape, but it shifts to the right. Renee, have you heard of this shift before?
Yes, sir.
What you call it?
The chemical shift. I think I learned this in A&P.
It's the Bohr shift. What is the Bohr shift? Oftentimes people describe it as a shift to the right. And it's because everybody has kind of looked at hemoglobin binding curves like this. Everyone is familiar with setting PO2 on the x-axis, binding on the y-axis, and at a different pH, at a more acidic pH, there's a slight shift to the right. So what does that mean? One thing it means is at this 50% mark, we've gone to a higher P50, all right? A while ago, at rest, the P50 was 28. Now the P50 might be 38. So if you describe the difference between how hemoglobin works at 7.4 and how it works at 7.2, you could say there's a shift to the binding curve to the right. You could say there's a shift to the saturation curve to the right so that the P50 goes from 28 to 38. All right. What does that mean, really? If you think about it as exercise, it sort of makes sense. What's going on is your hemoglobin is still going to the lungs where there's a high amount of oxygen and picking up oxygen, but it takes less of a drop in PO2 for that hemoglobin to deliver. Okay, so in this acidic situation, which is an exercise situation, hemoglobin is more apt to deliver. It's delivering here, right? Down here where it's not binding well, what that means is it's delivering well. So in a normal situation, it was still bound here. But in this exercise situation, since the curve is down here, that means it's delivering. Now it's delivering at P20, where a while ago it was still holding on at P20 at a partial pressure of 20. Okay, that's a shift down to 7.2. After a meal, you get another shift. you get a shift to the left after a meal, right? This is kind of like less talked about, but after a meal, the pH goes up to 7.6. After a meal, our blood pH becomes a little more alkaline, goes up. Colin, you know why that happens? After a meal, our pH goes up to alkaline.
I do not.
Annie, you know why our blood pH goes alkaline after a meal?
Does it have something to do with glucose release?
It might, you might know more than me, but this is the way that I think of it. You know what happens when you eat a meal? Your stomach is pumping acid into the stomach. So it's kind of like the protons that you have in your system, they start going in your stomach. And since all the protons are being harvested to go into the stomach, the blood is kind of now it's lacking protons. So it's kind of like lacking acid. So it becomes more alkaline. It's because all the acid that we have goes to the stomach. The blood in response is slightly more alkaline. Okay. And so this shift to the left is sort of what leads to us feeling a little sleepy, perhaps. Okay. Instead of delivering oxygen to the tissues, right, we're holding on to oxygen a little bit longer. So it makes you get kind of sleepy, you know, and makes you want to take a nap. So some of that stuff that where people talk about after a big Thanksgiving dinner, you want to take a nap, some of it has to do with this shift of the saturation curve to the left. The Bohr shift is the one to the right. There's not as much as a famous name for the shift that goes to the left, but I want you to understand that one as well. Okay. So that's some of the math behind how hemoglobin works. If I had to draw it, I could draw it as well. This is my alpha, beta 1, beta 2, right? When this hemoglobin gets to the lungs, there's a whole lot of partial pressure of oxygen. And so there's an oxygen here, an oxygen here, an oxygen here, and oxygen here. But as this hemoglobin goes through the circulatory system and gets to the pinkies, boom, it's delivering these oxygens. Right? It's not always necessarily so that it gives up every oxygen, but it delivers a lot of its oxygens. Okay, so now it's down at the bottom of the saturation curve. It's at the tissues where there's low PO2. Where there's low PO2, it tends to release its oxygen. Then when it goes back to the lungs where there's high PO2, now I'm at the high part of the sigmoidal curve, it picks up more oxygen. Okay. All right. So I want to talk to you about the fetus. We talked about the fetus already in its special circulatory system. All right. There's a lot of things about a fetus and a child that are similar, right? A fetus is an unborn child. A child after parturition, it's got arms and legs and eyes, and its heart's working. A fetus before parturition or birth, it's got arms and legs and eyes, its heart's working. But a child after parturition has lungs that are working. A fetus before parturition, it has lungs that are maybe being made and they're being formed, but the lungs are not working because that child is not taking a breath. Okay, so I know you know all that already. So this is review, and we went over 2 anatomical ways that fetal heart works differently. That first one is that there's an open foramen ovale that's open in the fetus. All right. That's the foramen ovale. In the fetus, it allows for a bypass of the lungs because blood comes into the right atrium and boom, it goes straight to the left atrium. It's kind of skipping this right ventricle, skipping the lungs. After birth, that foramen ovale becomes a scar. It's called the fossa ovalis, right? These two terms are kind of important, so I want you to know the difference between them. You know, if you accidentally mix them up, everybody knows what you're talking about. But in a class, on an exam, it's important to be able to know which one of these is open. The one that's open is the foramen ovale. That's what you find in the fetus. It's an open window. The one that's closed is the fossa ovalis. It's not a foramen, it's not open, it's not an open hole. The fossa scars over and closes this up because in a normal adult heart, you don't want that open right there. All right, that's the foramen ovale and the fossa ovalis. You remember the other anatomical trick, and that is we have the pulmonary trunk here. It's going to the lungs. A little bit of blood comes into the right ventricle, and a little bit of blood has to go to the lungs because the lungs are growing and they need a little bit of nutrition, and they need a little oxygen from the blood. It's just that the lungs themselves are not inflating, but we're building all the little cells here, so these guys need a little blood. So, okay, this right ventricle, it is pumping, and a little bit of blood goes this way, goes through that pulmonary semilunar valve, but we don't want to send too much there. And so there's this shortcut, kind of a cutoff. Anatomically, what you usually call that is an anastomosis. There's an anastomosis between the pulmonary trunk and the aorta so that we don't send too much blood to the lungs. Instead, if there's too much blood coming through the pulmonary trunk, we can send it to the aorta through this anastomosis right here. All right. Mr. Jones, you remember the name of this shortcut between the pulmonary trunk and the aorta? Mr. Michael Jones?
The ductus arteriosus.
Yeah, you know that good. All right, that's the ductus arteriosus. We already talked about that. And when it scars over, it becomes the ligamentum arteriosum. Ligamentum arteriosum. Ductus arteriosus is open. It transforms into the ligamentum arteriosum on something like an adult heart. Sometimes you can kind of notice that the pulmonary trunk and the aorta, they're kind of connected, and where they're connected, it used to be the ductus arteriosus. Now that just scars over and closes up because you don't really want that open in an adult. On the other hand, the alligator that takes a dive, it likes to keep using a connection right here. That connection in the alligator is called the foramen of Panisa, right? It's not exactly the same as what we find in a mammal, but functionally it kind of works the same way because it allows an alligator to shunt, do a P to R shunt, pulmonary to, no, a P to S shunt, sorry, from the pulmonary system to the systemic system so that it shunts away from the lungs and keeps the blood going to the systemic circuit. So these are two anatomical tricks that fetal heart can do. And then there's this biochemical trick. So some of y'all probably already know what I'm talking about, and that is in a fetus, these beta subunits are not made. A fetal circulatory system has a different tetramer. It's got 2 alphas and it's got 2 gammas. The fetal hemoglobin has this different tetrameric structure, 2 alphas and 2 gammas. We can number them the same, gamma 1 and gamma 2. Chloe, you know why? You know why there's 2 gammas? Say it again. You don't know why? Okay. It's because there's a contest going on here between the mother at the placenta and the fetus, right? The mother's blood comes to the placenta. The fetus blood comes from the placenta. The mother's blood has oxygen. The fetal blood comes in with the less oxygen. So what makes the mother's blood release oxygen, right? There's kind of like a competition for the oxygen. How does the fetal blood steal that oxygen? It's because this mother's blood has alphas and betas, but the fetal blood has alphas and gammas. And the gamma subunit has a stronger binding capacity. The gamma subunit allows fetal blood to take that oxygen, right? And it's not like these two hemoglobins, they're not actually touching. So it would be wrong to think of it like that. But the oxygen that's bound to this maternal hemoglobin, right? It's bound, it's in the pocket, it's bound, it's in the pocket. But this gamma subunit has a stronger affinity for that oxygen. So when this oxygen is on, off, on, off, on, off, it's more strongly bound by this gamma. So the fetal circulation can take away that oxygen. Okay. As soon as the fetus is born as a child, you start losing this gamma and it switches to beta. Okay, so that's a biochemical strategy that the fetal human uses. You got two anatomical strategies, ductus arteriosus and the foramen ovale, and this biochemical strategy of having gamma subunits that allow it to take oxygen from the maternal blood. I think that's a pretty good trick. Now, most of the story of hemoglobin and its structure function relationships almost in there for me, but the gamma subunit is famous now because of what happens during sickle cell anemia. Okay, sickle cell anemia is a disease caused by a mutation in hemoglobin. So it's this tetrameric structure that we've been talking about. In a sickle cell patient, there's a mutation in the structure of the beta hemoglobin that makes a slightly different structure. It sets up within red blood cells in a slightly different way, so that the normal red blood cell kind of looks like an inner tube, but in these patients that have a different beta subunit, their red blood cells set up like sickles. And this impedes circulation a lot. These sickles tend to, instead of bounce off of each other like these red blood cells here, like, these red blood cells bounce off of each other nicely. These sickle cell shaped red blood cells, they pack into capillaries, they get stuck, and it causes a lot of pain. And the tissues don't get their oxygen. So a recent gene therapy, practically a cure, is being used by turning off expression of the beta and turning on the gamma subunit in adults, right? In an adult, it doesn't matter necessarily, because we're not talking about having a baby inside of you. We're talking about turning on the gamma subunit while you're an adult. Well, this gamma subunit does not lead to sickle cell shape. If you have alpha and gamma, these patients, they get red blood cells that have their normal shape again. So physicians and molecular biologists are now starting to use kind of like an adult expression of the gamma subunit in order to treat sickle cell anemia. And it was done in one patient about 2 years ago, and she's healthy, and it's kind of in the South. I think she's either in Mississippi or Alabama, And it's a pretty cool gene therapy story because mostly gene therapy has not been all that easy to show success in humans, but this is one of the early cases where gene therapy is working in humans. Okay. All right. So I'll talk to you about this important respiratory pigment, which allows us to do our respiration. I wanted to take a step back though and talk about these respiratory organs. Now, if this was a nursing class, we would only be interested in what happens in human. But because y'all are biology majors, we want you to understand how respiration works in other animals. And another reason is that we come from this kind of animal. We come from an ancestral fish. So we would like you to understand how respiration works in the typical fish. Okay. You can stare at some fish that are in the hallway. Something like a bluegill fish or a bass. It can stay still in the water. And in order to bring water across its gills, the water comes in the mouth and the water moves out its gill chamber. And that's because what covers the gills is this bony muscular structure here called the operculum. That's why something like a brim, a bluegill or a bass or a sockeye can sit in the water and it doesn't have to move in order to bring water across its gills. It can use this operculum and do like this. We tend to call it gilling. It does, it opens and closes this structure. And when it does this, when it opens, that brings water in, okay? So the water is going in here, it goes across the gills and then out the operculum. So now we're gonna kind of like dissect this cartoon fish or use an x-ray, and we're gonna look underneath this operculum, okay? And most fish, most teleosts, most fish that we eat, not ancient fish, but fish living on Earth right now, have 4 gills on each side, okay? This is a gill arch. So there's a bony structure, a skeletal structure in the middle, right? And there's 4 of them. So the water is going past here and out here. So this is the skeletal structure in the middle. I want you to know what the gill looks like up close, right? So I'm going to add in what we got on this skeletal core. On each skeletal core, you could call it the gill arch. There's 2 sets of gill filaments. Kind of look like rabbit ears. But if you're really looking at a gill, they're real close to each other. I'm going to draw them kind of far away from each other just so you can see the structure, right? So you have 2 rows of gill filaments. So let's label this. This is the gill arch. It's skeletal. This is the gill filament. You can see that with your eye. But on the gill filament is some ridges. Now with your human eye, it's pretty hard to see these ridges. Now we're starting to get to a microscopic anatomy. And each one of these ridges is a lamella, is a gill lamella. Okay, so we're going from the supporting structure. The gill arch is a skeletal structure for support. The gill filament, got 2 rows of those. And on each one of these gill filaments, there's hundreds of gill lamella, right? So the blood is coming down this gill arch and going up through the gill lamella. So I'm gonna go in closer and closer at a higher mag. But before I do that, I want to show the other side of this gill arch. And if you look on this side of the gill arch, this is sometimes what you cut your hand on if you put your hand inside the mouth of something like a big redfish. And that's because on the other side of the gills, we have these gill rakers. And they're kind of there to point the food that also comes into the mouth, to point it to the inside to go into the gut, right? We don't want a big piece of food to come and hit the gills here. This tissue right here is kind of delicate. That's where oxygenation of the blood is going to happen. So instead, we have some more skeletal elements pointing on the inside there that moves food away from these delicate gill filaments and back into the esophagus, into the stomach. All right, so I'm going to go up close to these gill lamella. Go to higher mag. We have images in our textbook that reflect this. So let's say this is one big gill filament, and this is one lamella. All right. And if water's coming across this gill, kind of like we showed a while ago, right? The water is going through the mouth, across the gill, and then out the operculum. So the water's coming in this way. And we're getting close to the point of this drawing here. The way that blood goes through this lamella is in a countercurrent direction. The blood comes up here and it goes in this direction. Right? This sets up a countercurrent exchange that it's not like the fish has to think about this and design this for himself, but it's been designed by millions of years of evolution. And more often than not, a countercurrent exchange is set up in physiological systems rather than the opposite, which would be concurrent. A concurrent exchange. Okay, you hardly ever see that. If you see it, it's in a situation where the exchange of some kind of mineral or nutrient is not that important. More often than not, we see a countercurrent exchange. Okay, so you could think about the saturation of oxygen in the water of being something like 100%. Fresh water is coming into the mouth of that fish. It's got a whole lot of oxygen. When it passes over that gill, now it's lost about 50% of its oxygen. And what about the blood? The blood's coming in at something like a PO2, very low, of 5 millimeters of mercury. When it leaves the gill, we want it to have harvested oxygen from the water. Okay, so what do we want? We want it to have something like a PO2 of 50 or more, right? So this is what's going on at the epithelium of the gill. Blood is being brought in at a very thin layer of epithelium, and that hemoglobin that's in that blood is trying to harvest oxygen from the water. So I'd like for us to look at this even more closely. I'd like for us to compare a countercurrent exchange system to a concurrent exchange, and why this makes a difference. All right. So first, let's do this with a concurrent exchange system. So let's say that the blood is coming in this way. It comes in at a PO2 of something very low, something like 5 millimeters of mercury. This is the blood. I'm going to try to color it kind of red for you. Okay, The water's coming in at a high amount. This is concurrent, coming in the same direction. This is concurrent. It's confluent. That means the water is moving in this direction and the blood is moving in this direction, right? Let's say the PO2 of water here is 100 millimeters of mercury. Okay, this is coming in at 5, this is coming in at 100. There is sort of like a pressure. There's more oxygen here. And so if this is 5 and this is 100, you begin to increase the PO2 of this blood. It shoots up to 20. And this one is down to 80. There's still a gradient here. 80 pushing to 20, boom, this jumps up to 30. This goes down to 60. Okay, there's still a gradient. The PO2 is 60 here. It's just 30 here. So as this water comes close to this blood, boom, it drops from 50 in the water and shoots up to 50 in the blood, which is good. But now this concurrent system, the gradient has kind of bottomed out. It's 50 here and 50 here, right? So this water, it's like it doesn't have the head. It doesn't have the partial pressure gradient to push more oxygen in. Okay, so this blood is leaving at 50. This water is leaving at 50. There's no more exchange. Now, blood picking up a PO2 up to 50 is enough to like survive, right? This blood has oxygen in it. But what happens if we turn this around and we put the blood in a different direction? Let's say that the blood is moving in a countercurrent direction. Okay, the water is moving this way, the blood's coming in this way. Blood still comes in at a low amount. PO2 equals 5. Water still coming in at a high amount, 100 millimeters of mercury. Right, this is the blood here. Okay, we can pick up where we left off a while ago. Even if at this middle point here, there's a PO2 of 50 millimeters of mercury. As this blood is passing through, if this is at 100, now there's still a gradient here. So you could say that this might be at something like 60. It continues to push, right? Got 60 here. It's 80 here. Still continues to push. Maybe you bottom out at something like 75. Now this leaves at 75. And this drops at 75. All right. So let's fill in the numbers. As this water comes in, it's losing its oxygen. This oxygen comes in from the blood. It comes in very low. So even though there's a low amount of oxygen in this water, there's enough to push in about 10, 20, 35, right? So if you follow this, as the blood comes into the gills in this countercurrent system, even though there's only a low amount of oxygen in this water, it's enough to push oxygen into the blood. And because we're in a countercurrent exchange, as the blood is leaving, the blood that's about to leave the gill is exposed to the highest amount of oxygen in the water. So instead of bottoming out at 50, this water with a PO2 of 100 still has a gradient to keep pushing oxygen into this blood. So the effect is a PO2 of 75 millimeters of mercury. Okay. So having that countercurrent exchange has allowed for sort of like an efficiency. It's allowed for a higher capacity just from this anatomical difference to push more oxygen into the blood. So in most gills, we see it like this. We see it set up as a countercurrent system where the blood is moving in an opposite direction of the water. All right. That makes sense. Now, what's sort of weird about our human body is, okay, we come from animals that used gills. We don't use gills. Instead, we use lungs. And there's a couple things that are different in our lungs than what we just looked at. It's a pretty different situation. One thing that's different is that whereas the fish has a hole for water to come in and it's got a gill for the water to go out, that means a fish can always be exposed to fresh water with a fresh amount of oxygen. Whereas a human takes in air in one hole and he uses the same hole to push the air out. It's almost like from a design point of view, this is called tidal ventilation. It's tidal because kind of like the tide, it goes in and comes out. We inspire air and we expire air. Goes in and out of the same hole. It's like there's an inefficiency there, right? It comes in through our trachea, goes to our two lungs. When it comes to our lungs, it goes from our trachea into bronchi. The trachea split into bronchi. The bronchi split even more into little bronchioles. And these finally end in what often are called these blind end sacs called the alveolus. So this is where blood and air come close together. Now, the blood does not really get exposed to the air, right? The blood is in capillaries and it's close to these alveoli, and there's a thin epithelium between them. So at this functional level here, the gill is kind of similar to the lung in that in the gill, you have a very thin epithelium. The water does not mix with the blood, right? But the water comes real close. There's a thin epithelium and oxygen is taken away from that water by the blood. Once again here, there's a thin epithelium. In a lung, it's called the mesothelium. And blood is not exposed to air per se, but the blood is passing right on this back side of this epithelium and the air is on the front side. It's enough so that blood with the red blood cells can harvest that oxygen. Okay. So why can we exist with this sort of low efficiency tidal ventilation? Well, one reason is because there's just so much more oxygen in the air. If we had to design this in a low oxygen situation, like maybe in the future, or if we were going to make some humans that could be like Aquaman and breathe water, right? The way you would want to do that is to kind of like have an external vent down here so that you could always take in the air. Or maybe if we wanted to be humans that would fly, we would have to increase our efficiency by taking in air this way and sending out the CO2 this way. But we don't have that. Instead, we make use of these blind end sacs that take in air and then expire air. What that means is sort of like at the bottom of these lungs, there's always a residual volume. A residual volume of old air. So it's like from an anatomical point of view, the lungs are less efficient than the gills. They're less efficient because we always have this residual volume. You know, we think of the lungs as kind of like balloons, but they really don't deflate. Like when you breathe out, your lungs don't get small like this. Our lungs retain their size, and a lot of the air has left them, but because they retain their size, we have old air in there. So when we take new air in, it kind of mixes with that residual air. But it doesn't matter because there's so much oxygen in the air. So we kind of get around that struggle of having this inefficient sort of anatomy because we have such a high level of oxygen. Okay, so that's the difference. One of the differences between how the lung is built and how a gill works. We're going to talk about some modifications of this because we don't fly, mammals don't fly, but birds do fly. So birds have something similar to the mammalian lung, but they get a higher amount of efficiency out of their lung. So we're going to talk some about that, and we're going to talk about some other aquatic organisms that respire without using gills. And those would be some insects. Okay, I'm going to see half of y'all at lab today. That's it for some introduction to respiration. And if you have any questions about what we went over, you can email me, and I'll be seeing half of y'all later on. Okay, that's over and out for today. Thanks.